Okay, so yeah, I guess it's obvious that I come with the genetics expertise, but really what we're using the genetics for here is simply a tool to track or link fish across generations. So the context, I think it was spread no one in the room that salmon are declining, and when we think about the kinds of management intervention that humans have and continue to undertake, population stocking or supplementation have been popular approaches that we feel that there's something we can do to change the trajectory of these declines. And there's a variety of ways that we go about this. Traditional stocking or the SAS program that's been proposed here for the Miramichi that I'd like to think everyone's somewhat familiar with. I don't really want to talk about that. I want to talk about what a lot of people in the past have not been talking about, and that is, regardless of what we do, how do we know that it works? How do we go out there and assess the return on our investment? And by and large, the vast majority of this that's been undertaken, this is a component that's been missing. I think this is something that we've really got a strategy to try and address within what the CAST is trying to do. So, possibly one of the reasons that assessment is a little bit difficult is that we need a way to be able to tag supplemented fish. And ideally we want to be able to quantify the fish that we put in the water's contribution to future generations when we're talking about population supplementation for rebuilding or recovering populations. And the scale of this problem is enormous on two levels. First of all, a lot of the stocking that's been done has been on small fish. And so the kinds of tags that we can actually put in small fish were limited there. And the other side of the thing where scale becomes a problem is, this is some data courtesy of Ruth Whiffler at Pacific Biological Station. This is the BC Salmon Enhancement Program for several species of Pacific salmonids that are stocked. This is annually from the late 1970s to almost today, 2016 or something like that. And it's, the figure's a little bit small, but we're talking about hundreds of millions of fish a year that are being released for chum and sockeye in particular, but we're also talking about even in tens of millions of fish, Chinook and coho. And so we're talking about, this is an enormous logistical problem of how do we tag all these fish? And the answer is, we don't. The strategy that's used so often on the Pacific, in the Pacific Northwest is to use coded wire tags. And so coded wire tag is a little tag that bears a number. And this would be the size of the tag. These are injected into the snout of the fish. And typically some sort of external marking is given, so a clipping of adipose fin. So when an adipose clipped fish is captured at some point, whether it's an intercepted fishery or whether it's at a monitoring station on a dam or wherever it happens to be collected, the fish can be sacrificed, and some poor soul has to sit in front of a microscope and read tags. And this has become somewhat successful to the point that U.S. Fish and Wildlife has invested in a ton of these trailers with automated systems. So each of these rigs here can process tens of thousands of fish, automatic size sorters, and they coded wire tag. This is across different jurisdictions, and we're talking again about tens to hundreds of millions of fish annually that are tagged. So this is including, for instance, Columbia River and other rivers in the U.S. And so even with these enormous tagging operations, the problems with this are that low rate of tag recovery. If we compare the amount of fish that have been tagged to the number of tags that have been captured, we're talking about less than 0.5% of all tagged fish are ever recovered. And this leads to a real power problem in terms of how do we assess things. Also, in particular, also this requires lethal sampling. So we catch a fish as it's migrating up and we whack it off. We have no idea. It clearly does not contribute to future generations. So this is also labor intensive. And in the context of the SAS program, this is going to be problematic because we don't ever handle the juvenile fish. One of the benefits to a smolt-to-adult supplementation process is that fish are spawned naturally in the wild, rear naturally in the wild, and migrate as truly wild fish with no captive experience at that stage in their life. And so, a strategy that's becoming more popular to assess conservation stocking programs and these kinds of things is something called parentage-based tagging. And this is based on principles of Mendelian inheritance, just the fact that parents pass on their genes to their offspring. And so if we have a hypothetical set of parent fish, we can collect genotype information for all of the parent fish. And then when we capture an individual that we're interested in who and where they came from, we can generate the data to assess the genotype of this fish, and we can make some comparisons, and we can identify incompatibilities. So if the genotype at the first genetic marker for this fish is GG and this parent has AA, that's not compatible. So we can eliminate this fish. And likewise with this fish, and through a process of elimination, and some fancy statistical techniques, we can narrow it down and we can say this is this mom and this is this dad, and these fish came from wherever, and now we know who this fish is. Oops. So generally, what I've described to you, the process is in the context of the SAS program, is that we collect a small fin clip. The nice thing about this too is when we catch for this juvenile fish or adult fish returning, we take a small piece of fin. It's non-lethal sampling, and so this fish can go on and live its life, but we can still tell who it was and where it came from. So in the context of the SAS program, we collect fin clips for all of the adults. We generate their genotypes, so we effectively know the identity of every SAS fish. And if they go out in a hypothetical release, produce offspring, all we have to do is we collect a fish from the river. It's already tagged with its genetics that it inherited from its parents. So we do this, and we simply query a database, and we know who this guy is. So in order to pick as the most efficient, best set of genetic markers, we need to have some idea about the variability of these markers. And one of the other work that I undertook when I first came on with CAS was to conduct a population genetic study across the Miramichi Basin. So we have a really rich data set representing the 700 fish at 5,000 or 50,000 locations throughout the genome. And so I sort of sifted through the data and picked the SNPs. Well, I'm going to show you the process of how I picked the SNPs, but that we're really most interested in the ones that are most variable in the systems that are relevant for the SAS program. So at the time, the Little Southwest, the Northwest, and the Northwest Mill Stream. And so in figuring out how many SNPs do we need to develop a good parentage tool, I did some simulation work. And so we simulated based on realistic data that we have from the system. So we're looking for a minor allele frequency. This is the less common allele of at least 30%. We want it to be highly variable between individuals, so it gives us more power. If you only have the alleles present in 10% of individuals, then less than 10% of individuals will carry that in a subsequent generation. It's not going to be informative for excluding that for 90% of the fish as a possible parent. So simulated a hypothetical population of 10,000 parents. Let's say this is the number of fish in any one of these rivers. We took randomly grabbing 2 of these parents. We created, we did this 2,500 times to create 2,500 random babies of known parentage. And we essentially forget 90% of the parents, simulating that, okay, this is a 10% SAS contribution or SAS release in a somewhat pessimistic scenario. And then we use the genotypes that were of these parents to assign these offspring back to them. And so the figure that you're seeing here is an increasing number of SNPs in the panel from 100 to 400. And then the percentage of assignment success shown on the y-axis, number 1, with the green boxes. And on the secondary y-axis here, we have the number of misassignments, and they're represented by the red bars here. So what you can see is that even with 100 SNPs, we're getting, somewhere around 90% successful assignment, and this levels off up in the high 90%. And when we're talking about the number of misassignments, so these are fish that were assigned to a parent that's not truly their parent. We have this one weird issue with 300 SNPs, and where we get somewhere between 2 and 3. And this is actually just a quirk of the data. When I was running these simulations, I actually threw in the real genotype data from the 700 fish that we collected. And so this is actually more of an artifact of one of my simulated parents is very similar to an actual real fish that we caught in the river. And so these are misassignments to fish that weren't included in the simulation of babies. And that's not something that we would expect to actually happen in the wild. So this is with known SAS parent pairs. But what if we have a SAS mated with a wild fish? And so in this situation, this would be a known parent included in our group of parents, but another parent from the other 90% we don't know. And so when we run this, it's the same sort of figure. We can see as soon as we're hitting about 300 SNPs in our panel, this is plateauing out at 100% successful assignment with 0% misassignment. And so 300 is going to be a sufficient number of SNPs to give us 100% power to assign a random juvenile fish to SAS parents if it was truly sired by SAS parents. So now we have to go to the real data that we have and pick what are the actual locations in the Atlantic salmon genome that we're going to survey to look at this. And so we start off with this figure is depicting the data that we have across the 29 chromosomes from 1 to 29 of the Atlantic salmon genome. And I'm showing the minor allele frequency. This is how frequent the less common allele is. Necessarily this stops at 50% because if you go over 50%, it's no longer the less common allele. And so we can, maybe not so easy to see, there's a slightly higher density toward this lower proportion. This is simply a histogram showing the same distribution here, where we can see that we get slightly more common to have less common SNPs. And this is, we normally, in a natural population, if you were just doing sequencing to characterize this variation, we'd see a steeper drop-off, but the tool that we used in the first place is actually enriched for markers that are informative for Atlantic salmon, or for North American Atlantic salmon. So, we first want to exclude all of the markers that fall in the lower, the left-hand tail of that distribution. And so we grab everything that's got a minor allele frequency greater than 30%. So we gray this out. Now, we want to make sure that if you have two SNPs that are side by side, because the DNA is a linear piece of a thing, and if you have 2 points that are on a piece of string that are right beside each other, they don't really tell you they kind of tell you the same thing. They don't give you independent information. So we want to filter out things that show high levels of linkage, and we assess this by pairwise correlation between markers that are right beside each other. And when we do this, we filter out a whole ton more. But we're still working with quite a few thousand SNPs. And so finally what we want to make sure we're picking locations in the Atlantic salmon genome that conform to the way that we expect inheritance to happen. So these are things that we're not experiencing selection, that are segregating normally, and inherited in a predictable manner. And so we filter based on Artie Weinberg. For those of you from high school biology or introductory biology and hated genetics, you'll remember this well, or maybe you really try to forget. So we filter by that, and we lose a few more SNPs, and we're down to a few hundred SNPs, from which basically pick the remaining few to be reasonably evenly distributed across the genome, so we're not biasing to a particular chromosome or anything. And so we end up with a set of 500 candidate SNPs. And so now we have some, we can move forward with a real test of our panel. And I'm going to show you results from two situations where we've used this. The first is a lab experiment. Some of the work that David is going to talk about in a little bit. These are where we have crosses that were done in the hatchery to assess fertilization and survival of SAS versus wild fish, and fish that I'm actually using for some of the work that I'm doing going forward. And then the second is actually from the experimental release of SAS fish, to the experimental river in 2017. So from the lab experiment, we have almost 500 fish that we expect to be from the mortality crosses. And of those fish, 90% successfully assigned to mortality parents using that cross. 7% of them assigned to adults that were used in the fertility experiment. This is not surprising because these fish were kept in the same troughs, and with the experiments done in 2017, there were some issues with fish escaping their little boxes in the troughs. And so it's not surprising that we have a few escapees caught up in fish that we use. Finally, there's 3% that we have classifying as unknown. What's likely the situation with these is that we have one or two adult genotypes that failed during the genotyping process that we will need to redo. So it's likely that these individuals will be assigned to known parents. It's also possible that these individuals actually, if they have a high level of missing data in their genotypes, that we just don't have the power, and they accidentally misassigned to something else. And so we need to screen the data, and maybe their genotypes need to be rerun. What's nice about this, though, is that we have known families where these individuals were crossed in a known way, and so we can use that information to test the SNPs that we have in our panel, whether or not they're actually showing Mendelian inheritance, and whether or not we have a high rate of genotyping inheritance. This is actually going to allow us to whittle these 500 SNPs down to a set of 300 SNPs that's going to give us the best ability we can to do this parentage-based design. So David will talk a little bit more about this work, but in 2017 we were permitted to release 20 males and 20 females into the experimental stream, and we know that these fish some of these fish spawn successfully, and we went out in 2018 and did sort of a haphazard sampling. It's not quantitative by any measure, but we collected 111 young of the year from the mill stream, as well as 50 age 1+ par. And we were able to use the genotypes of the males and the genotypes of these young fish to assess the contributions to spawning in the mill stream. And so this figure showing the results of that. So what the figure is showing you is a count of the number of offspring assigned to a particular female represented across the bottom. So the SAS females are the first 11 to here, and then we have 6, I'm calling them unknown at the moment, females. I'll explain that more in a second. Each of the colors represents a different male contribution. So each of the blocks represents the relative number of offspring assigned to that particular male. The darker pink colors here that you can see reflect the SAS males. And all of these other colors, I've labeled them par or other males. They're in lighter color here. The reason that I'm calling these par, we know that par are an important contributor to spawning events in the Miramichi River. And there was a fence that should have prevented adult migration into this river for the vast majority of the migration period. However, at the very end, the water level got too high, the fence had to be taken out. And so these fish could, these unknown females could represent other females that came in after the fence came out. They could also potentially represent very small, I don't think we aged these fish. These could represent really small 1+s that essentially accidentally that would have been offspring of a previous cohort and shouldn't necessarily be included as younger here. David may have a comment to clarify on that. Well, except that we know that these two fish here actually were sired by a SAS parent, so maybe they are actually naturally migrating wild females. Anyway, the take home from this is that we clearly sampled very close to where female 13 had her nest because we captured 33 individuals. But clearly many of the SAS, more than half of the SAS females and half of the SAS males successfully reproduced. David is going to have some comments on why we don't have all of the fish. And for the most part, the male SAS are assuming a large contribution to the offspring of the matings. And just to give you an idea of what wild natural spawning in the Miramichi River can look like, this is some work done by Laura Weir in 2010 on Catamaran Brook. And so each of these pies represents a different nest that they sampled the eggs from. And the different slices of pie represent different male contributions to each of those nests. And so our results here for the SAS look fairly similar, where a couple of males are dominating the contribution, and then there's a successive slices of the pie that we assume is a precocious contribution. So, how many are on time, Tommy? Here. I can leave it right here.
Yeah, leave it for now, and we'll come back to.
Cool. Sounds good. So anyway, these are the groups that have contributed resources to this project in particular. I'd be happy to take